Pathogens are found in almost every place you can imagine, and they can cause serious harm to both humans and animals. Today we will be giving a PSA on 3 well-known pathogens: influenza, E. coli, and salmonella. E. coli and salmonella are both gram-negative bacteria that have a cell membrane, a thin peptidoglycan layer, an outer membrane, and a periplasmic layer.
The influenza A virus contains RNA as the nucleic acid, and it's encapsulated by the capsid and is surrounded by the lipid envelope, which contains the spikes.
According to the Center for Disease Control and Prevention, or the CDC, 13,000 people are affected by the 0157H7 strain of E. coli per year in the USA. In terms of salmonella, the CDC stated that there are around 1.35 million illnesses per year, with a total of 26,500 hospitalizations per year. More alarming, there are around 420 salmonella-related deaths per year in the USA.
According to the CDC, since from 2010 to 2018, there has been 9.3 million to 49 million illnesses of influenza. In addition, there has been 140,000 to 960,000 hospitalizations in that time frame. Even so, there has also been 12,000 deaths to 79,000 deaths annually from the influenza virus.
Salmonella and E. coli are both alike in that they have an almost identical cycle of transmission, which you can see in the diagram on screen right now. Both Salmonella and E. coli can be traced to an animal's various components. We can both get Salmonella and E. coli from milk that is raw. Additionally, we can get Salmonella and E. coli from meat that is undercooked. Feces from cows can spread Salmonella and E. coli to crops, which can then infect those crops, and once we eat those crops, we can be infected with Salmonella and E. coli. In addition, the feces can infect the groundwater with which we would water our plants or crops, and the same thing will happen. To prevent being infected with Salmonella and E. coli, you should take the following measures. Refrigerate any meat you might have, and when cooking it, make sure to cook to a proper temperature. Additionally, do not cross-contaminate, in that you should not place cooked meat anywhere near uncooked meat. You should also properly wash any produce you buy at groceries. The final method is to properly wash your hands with soap and water and scrub for at least 30 seconds. On top of that, you should also use hand sanitizer afterwards.
In terms of the influenza A virus, the virus can spread from marine animals to wild birds, from where wild birds can spread it to domestic ducks or bats. The domestic ducks can then infect the pigs through feces or other means, and the pigs can infect the poultry, and the poultry can then infect the humans, in which the poultry can also infect the horses, dogs, and this can all turn around to humans, and humans can be infected either from the poultry, the pigs, or even cats, and even human-to-human contact, or also aerosol or droplets. In order to prevent the influenza A virus, there are multiple prevention strategies that can be implemented. For example, going from domestic ducks to poultry, some of the prevention methods that we can use are indoor raising of the domestic ducks, monitoring, and also water treatment. In the step for human-to-human contact, we can use surveillance system, vaccination, in which masks can be used, and also quarantine using an effective surveillance system. From poultry to humans, the most efficient prevention method is decontamination and quarantine.
Current research being done in the field of E. coli and Salmonella research is being done in different packaging for produce and meat, bacterial detection kits, lactic acid bacteria, and microwave plasma processed air. Things that could be done better, that would require more research being done, is in the quality assurance of programs at farms, and agricultural training for produce farmers.
Current research to prevent the influenza A virus are the following: a universal influenza vaccine, global and national surveillance systems that recognize patterns across the world using computer modeling, aerosol or droplet transmission research, and also seasonal flu monitoring.
That's all, folks.